Assalamu alaikum, this is Saba Yousaf and I am a graduate of Nutrition Sciences from Pakistan. Today I am here to represent my project at 34 Scientific Meeting 2021 organized by Malaysian Society of Pharmacology and Physiology. My topic of presentation is mango seed copper oxide nanoparticles, its preparation and applications. These are my team members with whom I did this project in our lab, and we are affiliated with School of Health Sciences, University of Management and Technology. These are the table of content that I would cover during the presentation. First of all, as we all know, nanoparticles are a class of matter and they include particulate material of one dimension that is less than 100 nanometer minimum. They are of different classes of different shapes, depends upon their shape, their sizes, their physical and chemical properties. They are of different classes, like they can be copper based, they can be carbon based, they can be gold based, and metal and polymeric, etc. They have a lot of applications, like as I have mentioned, some biological sensing. They are used in capturing of carbon dioxide, gas sensing, and delivery of drugs. Copper oxide nanoparticles are widely used for different purposes and a very common class of nanoparticles, while polysaccharide that we have used in this project is extracted from plants like gum arabica and have been used to enhance the properties of nanoparticles for several years. The main objective of the project is the preparation of mango seed polysaccharide nanoparticles. We used mango seed polysaccharide as it is the cheapest source. Mangoes are cultivated in a large number in Pakistan every year, and they are very common and easily available during summer seasons. Material method, the whole process of preparing mango seed copper oxide nanoparticles was consist of two steps. Step one is the preparation of copper oxide nanoparticle, and step two include capping of copper oxide nanoparticle with polysaccharide, mango seed polysaccharide. The copper oxide nanoparticle preparation was done using 184.564 gram of copper sulfate pentahydrate as a raw material in 1 liter distilled water, and 1 molar solution was obtained of blue color. The color changes when we added ascorbic acid as a reducing agent. After that, it was filtered using Wortmann number 1 filter paper, and then it was dried in oven at 90 degrees Celsius for 1 hour, and we got a solid crystals of copper oxide nanoparticle. The box in the right shows different measurements according to the mole and gram that we used during the preparation. The step two was capping of copper oxide with mango seed polysaccharide. There was two samples, sample A and sample B. The difference between the two samples was actually of the ratio between polysaccharide and copper oxide. Sample A contains 1 gram polysaccharide with 0.5 milligram copper oxide, while sample B contains 0.5 gram polysaccharide with 1 milligram copper oxide. While the further process was same for both the samples, that the polysaccharide copper oxide ratio added in 50 ml distilled water with 0.5 mole citric acid. It was placed in shaking water bath for 1 hour at 75 degrees Celsius temperature and 175 PRM speed. After that, it was kept in an oven for 1 to 2 hours at 75 degrees Celsius for drying, and we obtained light brown and dark brown solid crystals of polysaccharide nanoparticles, and that was further used for characterization. We used UV spectrophotometer for characterization, and the range of the wavelength used was between 200 to 800 nanometer. The results of the preparation was as follows. This is a pilot study was done before the actual process. We used copper sulfate without any measurement in 500 ml water, and used lemon for extracting vitamin C, and we got 0.414 gram of copper oxide nanoparticle. This is the actual solution. Solution 1 contained 93.273 gram copper sulfate in 500 ml water, and 3 gram vitamin C was added using vitamin C tablet. The yield was 0.712 gram of copper oxide nanoparticle. While solution 2 contains same amount of copper sulfate with 500 ml water, the difference between the solution was due to different level of vitamin C used. We used 19 gram in the solution, and there was a difference between the yield. We actually wanted to know the difference between the yield depend upon vitamin C level, but there was actually not a big difference between the yield as it is shown in the graph. While the capping of the two samples was done, and there was actually a color difference between the two samples, as it is shown in the pic. Sample B contains a light brown solution, while sample B2 contains a dark brown solution. Characterization was done. First of all, copper oxide nanoparticle was characterized as it is shown in the graph. The peak absorption was 440 nanometer and it was totally in agreement with the previously published data. While polysaccharide, the peak value was 795 and it was not according to the previous data. While polysaccharide copper oxide nanoparticle, the absorption was increased due to copper oxide nanoparticle and it shows a strong bond is established between polysaccharide and copper oxide. In conclusion, the yield was changed due to vitamin C level. There was difference between peak levels and peak absorption of polysaccharide increased with copper oxide. These are the references according to which we did our project. Thank you. Any questions and queries are warmly welcomed.